Every day, your brain relies on cellular cleanup systems to keep billions of neurons healthy. But what happens when one of those systems fails? Let's find out. Here's our roadmap. We'll learn what alpha-synuclein is, how autophagy clears damaged proteins, why that matters for healthy neurons, how protein buildup contributes to Parkinson's disease, and why researchers are excited about PA200 and Blm10. Every cell functions like a tiny factory, producing proteins that help it communicate, generate energy, repair damage, and survive. Most proteins work as they should, but over time, some become damaged or misfolded. One of these proteins is called alpha-synuclein. Despite its complicated name, it's a normal brain protein. Healthy alpha-synuclein helps nerve cells communicate, supports dopamine release, and contributes to memory. Normally, alpha-synuclein performs its job without causing problems. But when it folds incorrectly, it sticks to other misfolded proteins. These clumps grow into larger deposits called Lewy bodies, disrupting normal cell function and placing increasing stress on neurons. These protein clumps do far more than take up space. They disrupt communication between neurons, impair mitochondrial function, increase inflammation, and reduce energy production. Every healthy cell has its own cleanup system called autophagy, or self-cleaning. Like a housekeeping service, it removes old proteins, damaged mitochondria, and cellular debris. Autophagy is one of the cell's most important protective processes. It removes damaged proteins and worn-out cellular components, keeping cells healthy and efficient. But when this cleanup system slows, waste accumulates. Your brain depends on healthy autophagy because most neurons are built to last a lifetime. Unlike many other cells, they can't simply be replaced. They rely on autophagy to remove worn-out proteins and cellular waste. As abnormal alpha-synuclein accumulates, it overwhelms the cell's natural protein quality control systems. Autophagy and the ubiquitin proteasome system can no longer keep up. Damaged proteins build, placing the entire cell under increasing stress. Once this vicious cycle begins, each step fuels the next. Less autophagy leads to more alpha-synuclein, more damage, and more inflammation, further weakening the brain's ability to clean itself and accelerating disease progression. As this cycle continues, neurons produce less energy, communicate less effectively, and repair themselves less efficiently. Over time, they become increasingly vulnerable to dysfunction and, ultimately, cell death. As neurons are lost, symptoms gradually appear. Early signs include tremors, slower movement, poor balance, muscle stiffness, sleep disturbances, and changes in memory or thinking. Parkinson's disease develops over many years. Abnormal alpha-synuclein often begins accumulating long before symptoms appear. During that time, protein buildup damages neurons and overwhelms the brain's cleanup systems. By the time movement problems become noticeable, many nerve cells have already been affected. One of the strongest findings in Parkinson's research is the presence of Lewy bodies. These protein clumps, made primarily of misfolded alpha-synuclein, are found inside many affected neurons. Their presence provides strong evidence linking abnormal alpha-synuclein to Parkinson's disease. Many Parkinson's symptoms result from the gradual loss of dopamine-producing neurons. As dopamine levels fall, movement becomes slower, less coordinated, and more difficult. Researchers then asked an important question. Can cells remove these harmful proteins more effectively? Instead of simply treating symptoms, they began exploring ways to strengthen the brain's protein cleanup systems. Researchers identified a naturally occurring protein called PA200 that activates the cell's 20S proteasome. Acting as the cell's recycling system, the proteasome breaks down damaged proteins. By boosting this process, PA200 may help prevent harmful protein buildup. Scientists often study yeast because it shares many of the same cellular recycling systems found in humans. Blm10, the yeast equivalent of PA200, helps researchers understand how stronger cleanup pathways may improve protein quality control. The results were encouraging. Increasing PA200 or Blm10 activity reduced abnormal alpha-synuclein buildup, decreased toxic protein aggregates, and improved protein clearance. Researchers made another important discovery. Even when normal proteasomes became less effective, PA200 activated proteasomes continued removing damaged proteins. This suggests an alternative cleanup pathway that remains active during cellular stress. Improving protein cleanup may reduce toxic protein buildup, helping protect neurons and support healthier brain function. Although these findings are still limited to laboratory models, they point toward promising future therapies. Because abnormal protein buildup occurs in several neurological disorders, researchers are exploring whether improving protein quality control could benefit conditions beyond Parkinson's disease. The future of brain medicine may focus on preserving the brain's natural cleanup systems before significant damage occurs, not simply treating symptoms afterward. Although this research is still in its early stages, it represents a promising shift toward protecting brain health at the cellular level. One of the greatest opportunities in brain health may be protecting neurons before significant damage occurs. Healthy protein recycling may help cells remove damaged proteins before they disrupt normal function. Let's summarize the key concepts. A healthy brain depends on healthy cellular housekeeping. Together, autophagy and the proteasome maintain protein balance, supporting healthy neurons and long-term brain health. When the brain's protein cleanup systems begin to fail, abnormal alpha-synuclein and other damaged proteins accumulate, placing increasing stress on neurons and making it harder for the brain to function normally. Taken together, these findings represent a promising direction in Parkinson's research. In laboratory models, PA200 and Blm10 improved the breakdown of abnormal alpha-synuclein and supported healthier protein homeostasis. The most important message is that healthy protein cleanup is essential for brain health. Research suggests that PA200 and Blm10 may strengthen protein cleanup pathways alongside autophagy. As we conclude, remember this: protecting the brain begins with protecting our cells. Every day, our cells remove damaged proteins and maintain the balance needed for healthy brain function. Research into PA200 and Blm10 offers a promising new direction, although the science is still evolving. By protecting our cells, we help protect the brain. Thank you for watching.